Good afternoon, everyone. I am Parsa, and today I have the honor to present Dr. Bijsterbosch today. Hope I pronounced it correctly. Okay, I practiced that. So, Dr. Bijsterbosch, I'm going to give a quick background of her. She has worked in brain imaging since 2007, following a PhD in Sheffield, United Kingdom, and postdoctoral experience in Oxford afterwards. And she joined Washington University in St. Louis in 2019 as assistant professor in the Computational Imaging Research Center of the Department of Radiology. Her research program develops computational neuroimaging approaches and leverages epidemiological datasets to identify brain markers of mental health and to elucidate principles of functional brain organization. In addition, Dr. Bijsterbosch is an advocate for open science, serving as handling editor at Imaging Neuroscience and prior chair of the Open Science Special Interest Group with the Organization for Human Brain Mapping. She's also engaged with teaching efforts as co-director of Systems Neuroscience Course in the Division of Biology and Biomedical Sciences, co-chair of the FSL Course Organizing Committee, and also the lead author of a textbook on functional connectivity analysis, which was published by Oxford University Press in 2017. So without further ado, please give a warm welcome.
Thank you. Thank you for a nice introduction. Thanks for having me. It's great to be here. We were talking earlier as we were setting up, and I'm happy for this to be super interactive. So, feel free to ask questions anytime I hear there's like a microphone that can go around. So put your hand up, and comments, questions, everything very much welcome. I'm happy to make this into a discussion. So as the introduction sort of mentioned, my lab does two different sort of types of projects. One is a little bit more applied on depression, and that's what I'm going to focus on today, given that we're in a mental health hospital. And the other side of things is a little bit more of a kind of basic science side, focusing on resting state functional connectivity and how we can get the most information essentially out of that data set. And so I won't touch on that too much here, but I'm happy to discuss that. And you'll see some sort of you'll see a lot probably of my, I'm half neuroscientist, half data scientist or data geek, I think. So that will probably come through. So these are some relatively older studies looking at both the structural and the functional neurocorrelates of depression. And a lot of this work is focused on limbic regions, default mode regions, frontal regions, subcortical regions like the hippocampus and the amygdala. And so if you look at some of the literature, you might think that we sort of understand the brain basis of depression pretty well. And I think mostly my talk is to tell you that we don't, is the long and short of it. So this is a study that we did when we first started the lab using data from the UK Biobank. This is too many words, and I'm not expecting you to read this. This is a canonical correlation analysis between lots and lots of brain imaging features, both structural and functional, and several kind of self-report measures of mental health broadly. And the good news is that we find significant associations, and they're pretty reproducible as well. So the red lines are the original kind of cohort analysis, and then the blue lines are from an independent replication cohort. So you can see not everything replicates, but you get decent replication. But these are all of the significantly contributing features, and this is only the structural figure. There's a figure that's even bigger, that's sort of the functional features figure. So the main kind of point that I want to make with this is that it's not as sort of neatly localized and sort of meaningful organizational principles as maybe those earlier pictures that I showed suggested. And in fact, I really like this study. I think this is by someone from Simon Eickhoff's group in Germany. So this is a meta-analysis, and these dots represent previous studies that found some association with depression, and you can see it sort of covers everywhere in the brain, and none of these are significant. So essentially, this is a null study showing that if you do a meta-analysis of existing studies, it's really hard to find like really robust findings. And that's where big data comes in. So this is a figure from Scott and Merrick's paper, the infamous BWAS paper from a couple of years ago, essentially showing a basic statistical principle of sampling variability. So if you have a relatively small sample size, you might get quite a variety of different results if you got multiple cohorts of that small sample size. And so you might get very strong positive results, very strong negative results. And then as you get larger sample sizes, your sampling variability goes down, so the variation that you see goes down. But the other big message of this paper is that the strength of associations between brain and behavior tend to be relatively small. And so my lab, and most of what I'm going to present today is using data from the UK Biobank, which I think many of you have heard of, but just to make sure we're all on the same page. So this is a cohort in the UK. It's not a disease-specific cohort, but it's also not a healthy cohort because there's no specific inclusion-exclusion criteria. And it's big. I mean, it's 500,000 people, out of which 1 in 5 is getting scanned. So I think at the moment we have about 60,000 scans of participants sitting on our servers. And that creates opportunities for looking at things like mental health and depression in a sort of more of a population perspective. So it's poor data compared to like very clinically specific recruited data where you've got much richer sort of clinical instruments, and that's something that we can chat more about. But it's rich data in terms of like avoiding some of the if you get money to scan 100 people, then you have to decide, do you get people who are on medication or not on medication, first episode or not? And so there's a lot of like decisions that you have to make in terms of exclusion criteria. Whereas here, you can kind of study mental health and all of its messiness, and you have some of the power to do that. So there's pluses and minuses, but that's kind of my perspective on it. Going back to that meta-analysis that we saw earlier, so this is work that we haven't quite completed yet, but I just kind of wanted to show a slide on it briefly. So these are volcano plots from it's called a mega-analysis. So we took 6 large data sets, including the UK Biobank, analyzed them separately, and then did a meta-analysis across those. The good news is that anything below the line, below those dotted red lines, is significant, and so we can actually find pretty significant associations with both depression on the top and neuroticism, so more personality trait associated with risk. At the bottom, the colors indicate different kind of networks, the Yale networks, if anyone knows, so like different big networks in the brain. And I think what we're seeing here is, again, a sort of whole brain pattern rather than these localized effects in particular studies of interest. So we're still digging into this. We're wanting to do a lot more analyses in terms of like structure function comparisons and sort of localization, spatial organization comparisons. So yeah, that's something that I'm excited to be able to tell more about at some point, but just wanted to show some of these results. So what I want to spend the rest of this this talk on is essentially thinking about heterogeneity in depression, and the idea being that maybe the reason why it's kind of hard and why the effect sizes are small and why the sort of effect sizes are quite diffuse in terms of different brain systems in depression is because people with depression differ a lot from one another. And so I want to go over a few different studies that try and dig into that in different ways. So one way in which there's heterogeneity or variability between patients in depression is in terms of the clinical presentation. So to be diagnosed with major depressive disorder, you have to meet 5 out of 8 or 9 criteria in the DSM, and so that means that these are 2 made-up patients. As you can see here, they both have the same diagnosis, but they only have one symptom in common, and that's a pretty vague one. I'm feeling pretty fatigued right now, to be honest. So this is a made up example, but this is pretty realistic given the diagnostic criteria. And so this is what the kind of one of the most obvious ways in which patients differ from each other in depression. But there's other ways as well. There's depression is an episodic syndrome, and so there's differences between state and trait. So someone's general propensity or risk of developing depression versus someone's current state, whether they are in a episode or not, and that those two things are usually quite highly correlated. Those who are at risk are also the most likely to be experiencing an episode at any one time. But that's another kind of more within patient source of heterogeneity. There's also comorbidity, so lots of different, a lot of patients with depression suffer from other things as well. We've discussed at the lunch today that a lot of patients with neurodegenerative disorders also experience depression. And so there's a lot of intricacies there. And so essentially, and then the last question is, are subtypes the solution? So essentially what I'm going to do in the rest of these talk is talk about 4 different papers that we've worked on over the past few years in our lab that kind of try and address to some degree each of these questions. And again, happy to discuss at any point in going to the details. Okay, so let's start with this question of clinical heterogeneity. There we go. So let's start with this idea. And this kind of I had a very talented PhD student, Kayla Hannon. She just defended. It's now Dr. Hannon. She just defended a couple of weeks ago. And we were sort of a few years ago, like trying to develop her project. She was really interested in this question of heterogeneity. And we were thinking like a lot of the ideas behind subtypes sort of assume a mapping between the brain and the clinical presentation. So this picture is kind of trying to visually show like an overall depressed population, some subtypes within it, and the assumption that if you get a cohort who presents similarly in terms of their clinical presentation, they might also have, distinct neurobiological or neuroimaging correlates. And that's kind of a one-to-one brain symptom mapping. So in that scenario, it doesn't matter whether you determine the subtypes based on the clinical information or based on the brain information, you should get the same answer, essentially. On the other hand, there might be this idea of kind of many-to-one brain symptom mapping, that even within people who present in the same way clinically, there might be different kind of mechanisms from a neurobiological perspective giving rise to that same sort of clinical presentation. And that's many-to-one. And these are not mutually exclusive. You might have some sort of subtypes that have a stronger one-to-one mapping and some that have many-to-one mapping. And so the purpose of this project was to essentially test both of these hypotheses, test whether we have evidence for one-to-one, and then test whether we have evidence for many-to-one as well. And so we did this in a slightly different way from how people usually approach this, and it's partly because we had so much data to begin with. And so we decided, instead of doing data-driven subtypes that are some sort of dimensionality reduction or clustering algorithm, in this case, we decided to do it in a really sort of feature-based, clinically based way. So we looked for people who were high in one symptom and low in the other symptom. So these are not subtypes, and they're, in a way, they're kind of unique patients, because most participants have experience a mixture of symptoms of depression. But because we started with tens of thousands of people to begin with, we were able to find those unique people who are like high in anhedonia but low in mood symptoms, for example, and so on. And so that is even more sort of clinically homogeneous than data-driven clusters or subtypes might be, because we're really dissociating things that are usually collinear or correlated. And we didn't just do that based on symptoms. We did that on some other dimensions of severity. So you can determine severity both in terms of chronicity, number of episodes. This is the UK Biobank. These are older participants, or in terms of like acute symptomatology. So that's that sort of state trait differentiation to some degree. And then, of course, late onset is sometimes thought to have, so participants who have no history of depression, but then once they get older, have first episodes, are sometimes thought to have a different kind of mechanism behind them. So, and then at the bottom here, we have a heterogeneous group, which is our biggest group, which is basically anyone who didn't meet criteria for anything else. So these are participants who, by the core standards of the UK Biobank, which is not a structured clinical interview or anything like that, but still sort of clinically relevant criteria. So these are participants who meet criteria for depression, but are sort of a mixture of all of these different things. And so the first question that Kayla, who led this, wanted to ask was, she used normative modeling, both on structural and functional and diffusion parcellated features. And so the normative model was trained on, I think, 8,000 healthy controls. And so the first question was, are the normative deviations stronger in these clinically homogeneous group compared to the heterogeneous group? And that's what you would expect if there's some one-to-one mapping, right? Because if you parse out sort of the clinical sources of heterogeneity, then hopefully we would find sort of bigger effect sizes, more different from normal sort of patterns. And then if there is some heterogeneity between these sort of clinical groups, then you might expect sort of different patterns between the groups. And so I'll show you the results. And there's a lot of information here. You can stare at this at a long time. We did. But I'm going to draw your attention to a couple of things. So essentially the colored bars are the different groups, and the dark red sort of most right-hand colored bar in each column is this heterogeneous comparison group. And so you can see that tends to be smaller or closer to zero than at least some, or in many cases, all of the other groups. So that's sort of confirming our hypothesis that when you parse in a very sort of tightly controlled way this clinical heterogeneity, you get larger effect sizes in the brain. And the other thing is that you can see sort of interesting patterns, sometimes even opposing directions patterns. So negative essentially means less than healthy controls, and positive means more than healthy controls. Yeah. So, sorry. Otherwise, people on Zoom can't hear you. So, I don't think
yeah, that's.
The heterogeneous group is an exaggerated group because you remove all those other subjects. And so if you put all of them together, the effect sizes that, have you looked at the effect sizes if you have all of them together? When you look at it should be somewhere in the middle, but
it's a good question. We did so many things in this. I'm sure we've done that at some point. And so another thing we've done, because the heterogeneous group is also much bigger than any of the other ones. So we did some bootstrapping as well within the heterogeneous group to see how variable it is. I think we always found that the heterogeneous group was smaller, and it wasn't as much in the middle as you might expect, because it stayed relatively smaller. Because I think to some degree, if you put all of these groups back in, some of them cancel each other out, right? Because they're in opposite directions. So it's not just that we've removed the most interesting people, but we've also split them up.
So that was the other question.
Yeah.
If you put these other peoples together, or everyone together, how correlated they are with each other? If we don't know, because you can afford this, but most people in their studies cannot.
No.
And when they cannot afford this, are they getting something completely biased that is not reliable? Or is it correlated at least, but the effect size doesn't reach where you are reaching?
I think we didn't systematically test quite that question, but my intuition is that it would be correlated, because to some degree, like, we're seeing amygdala, insula, right? So these are not unexpected things to find based on the literature. So I think we are finding something similar, and it would be correlated, but smaller effect sizes. Yeah. And I mean, the other thing is that the advantage that more clinical studies have is that they tend to get more ill people, more severe people, and I think that actually boosts the effect size a little bit. So you get a trade-off there.
Because that's interesting. This is the like they can't see these, but you can subset these for the severe ones, and you can kind of tell us if I was studying 100 really sick people from my group. Yeah, there are things you can see, but these are the only thing you can see versus that we have these. We can tell you what you can't see at all.
Yeah,
so
exactly. Yeah, and to some degree, the bright blue group, the acute impairment group, is I think the most similar to, because that is a subsample that is relatively severe at the time of scanning. So I think so. Yeah, those might be more reflective than the people in the heterogeneous group, which is more of a population mixture, I would say. Yeah. Okay, so the takeaway from these results is that we do actually find some evidence for this one-to-one mapping. If you parse sort of clinical variability, you get stronger effect sizes on the brain side. But we wanted to do one other thing, which was to test the hypothesis of many-to-one. Like, even though we see that, do we still find evidence for sort of neurobiological clusters? And what we did here was essentially do k-means clustering, but within each of these clinically homogeneous groups. So we know that these people present very similarly clinically, but when we do k-means on neuroimaging features, can we find kind of separable groups in that sense? And so, I mean, k-means is always going to give you an answer. It's going to comply. It says, how many groups do you want? Okay, here's that many groups, right? And so this is a plot of stability to try and test across bootstraps, how reliable are those subtypes that we get. And so you can see that in some groups, for example, the acute impairment, which is like the most severe group, that was the one that found the most stable sort of subclusters in a data-driven way. And so I'm going to focus in on that a little bit. So these were the two clusters that we found, which didn't differ that much in terms of severity or sex, or age, which is not super surprising because they were so homogeneous to begin with. And then we looked at sort of held out information. So in the middle here is a summary plot of the brain features. Now, these were the features used to determine the cluster. So it's not surprising that the clusters differ on this. We actually resisted doing this several times because we were like, no, that's a circular thing. You shouldn't do that. But reviewers made us do it. But it's kind of interesting because so the black line is like the healthy controls. So that's essentially like the 0 line when you're doing normative deviations. And we see one of the groups that actually across different modalities has relatively preserved neurobiology, and another group that has relatively worsened neurobiology. And then that maps onto cognitive scores. So the group with the preserved neurobiology actually had cognitive scores that were similar to normal healthy controls. And then the group with the sort of worsened neurobiology had reduced cognitive scores. And the cognition, that was an independent finding, so that information was never used in any of the definitions of these subgroups. So this is, I mean, ideally you would try for like, I'll get to you in a minute. Ideally you would try for like, treatment effects and things like that, but of course that's not something we can do with this cohort. But yeah.
So are the values in there like the mean deviation or like grade by volume, or is it making one thing?
This is averaged across regions, just so that you can get the spider plot. No,
so like across the entire brain.
Yes.
An average.
Yes, there's an average of reduction. Yeah. I think somewhere in I mean, this paper has one of those 30-page supplements. I think somewhere in there's the like more detailed plot for every region. But yeah, this was averaged. And so I don't think this is the final say on this, but what this is showing is that there is, at least in some of these groups, maybe some evidence that there's still kind of for this idea of many-to-one mapping, that there's multiple neurobiological pathways that might lead to the same symptoms and that might actually have an impact on some of the other aspects like cognition, which might be relevant. late life depression is thought of as a prodrome or a risk factor for increased risk of Alzheimer's disease. So we were interested in this finding from that perspective. We are unable to link it directly to that, of course, in a clinical sense, but it's supporting that literature, I think, to some degree. Okay, so that was all I was going to say about this study. I'm going to move on to kind of the state versus trait story. I don't know if there's any. I'm happy to pause if there's any more questions on that one, or come back to it at the end. Right, seeing none. Well, oh, it's to your other side.
Thank you. So, did you try to link the or look at the differences between two clusters and disease status? Like in your other slide, you had a number of 2,080 individuals in UKI. So looking at that, or ADPR as a score, for example, or
We didn't do that in this in this study, partly because when you get to the clusters, the sample sizes actually get pretty small. And we didn't want to compare things. We didn't want to run into shoot ourselves in the foot by getting too many multiple comparisons going. So even though we would have been interested in looking at a lot more things, we didn't in this study. But we are doing another study where we're explicitly looking at the link between neuropsychiatric symptoms and cognition, and there we are including the PRS too. In addition, in more of a we're not doing the subtyping there, so we're using the bigger data set and doing some structural equation modeling. I'm not presenting that yet. It's not quite. We're getting some interesting results. It's not quite ready for the limelight, but yeah. Cool. Okay. So this is work from Wei Zhang, postdoc in the lab, soon to be moving into a faculty position, hopefully. And of course the question here, so state and trait, current experience of symptoms versus generic risk are correlated, at least 0.6, 0.7, and this is real data from the UK Biobank. But if we think about it and so they're usually not really differentiated in the literature that strongly, because it's hard, right? If things are highly correlated, you can't really differentiate between state and trait. But if we think about it from like a, utopian world where we would have a clinical biomarker for these things, you would use a trait biomarker in a different way clinically from a state biomarker, because a trait biomarker, you could, scan a kid and see at what risk level they would be, and like help them with coping skills, for example, before the onset of the first episode. Whereas if you had a state biomarker, that's something that you would need to actually track, for example, efficacy of medicines, because you need something that can change as a function of that. And so we were interested in this question, and they're highly collinear. But again, when you're fortunate to have this many participants, you can kind of find the unique people, right? You can find the people who have a strong history of depression, but happen to, at the time of the scan, not report any depressive symptoms, or vice versa, who sort of report. Well, for the state, we actually looked at change in depression. So we looked at people who changed it in a naturalistic way across the 2+ years between the scans, went from an episode to not an episode, or vice versa. And so you end up, of course, with a much smaller sample size, but you do completely dissociate state and trait depression in a way that is much harder to. I mean, if we had to recruit for a study like this, or for the previous study, I'd be presenting this in 20 years' time, right? Because it would take a very long time. And to some degree, these are the sorts of questions that we're looking for with the UK Biobank, because I don't want to do a poor man's job at a research question that can be much better done in a more rich clinical sample. So it's these sorts of questions that I think that sort of population data is particularly well suited for. It's the questions that you couldn't even start to ask in other data sets. And so our hypothesis, my hypothesis was that we looked at functional data mostly, and I was thinking that state would be related to like functional connectivity between brain regions, something that changes dynamically over time, whereas trait might be reflected in sort of network topology, which tends to be a lot more stable over time. So this was actually talking about open science. We did some we pre-registered this. We even submitted it in a couple of places as a registered report, and so we had pretty clear like plans and hypotheses, and found nothing like that. So these are the results, which are puzzling in a couple of different ways. So these are for trait depression. So these are two visual networks, you heard that correctly, that have a stronger amplitude, so essentially stronger signal in participants with high trait versus very tightly matched controls. That's the other thing. We have lots and lots of controls, and so we can find people who are nicely, age, sex, and head motion matched to our patients. And this was very robust, that we did this at multiple different network dimensionalities, as we talked about the other day. And this was the finding that we had. And we didn't find anything for state. Now that might come back to the not being severe enough aspect of it. Yeah, go for it.
Have you colored the previous plot based on your 7 groups of the other study?
Oh, we have not done that. No. Interesting. What would your prediction be?
I think that severely impaired won't be less,
Yeah.
but for
Yeah.
I don't know much about depression. I'm just putting myself out there.
No.
But
No, I think you're right, and certainly not in because this is the box of the trait people. The state we actually define by change, so people with either a big increase or a big decrease, and you're right, those might not be the most severely impaired because the most severely impaired might be less likely to change over time. So
it would be interesting if some colors
Yeah.
would be separate.
yeah, that's an interesting idea. And actually, I think this is not a fluke. We've gone back. This is partly the reason why we're doing that big meta-analysis to test, and doing it in different Yale networks to test whether, to what degree the visual and somatomotor cortices are associated with depression. Because we have seen this in a lot of places, in a lot of data sets actually, and I've had conversations with other people and they've seen it as well. I think it's often either in a paper but not discussed in the discussion because people are like, I don't know what that means, or it's just ignored altogether because people are like, oh, we're going to focus on amygdala or hippocampus or default mode. So we're actually seeing that some of the strongest, most reliable associations are in like earlier visual and somatomotor cortices. I don't know, like at some point we want to test different hypotheses about why that might be. We've played around with it a little bit to see if it was maybe sleep related or related to somatic symptoms. I think there's a question over there. Whilst I'm chatting, you can head over. So I'm interested in hypotheses about what you think this might be related to. Yeah, go ahead. Oh, I don't think we broke it down by sex. I think we might have controlled for sex. I'm not sure. I'd have to go back. It's been a while. This has been out for a little while. So, well, it's been a little while since it's not been out for that long, but it's been a while since we wrapped it up. I guess things take a long time. So it's been a while since I was in the nitty-gritty for this. So I'm not sure. I'd have to check.
I work in a lot of depression and we see like effects that are in the visual areas are driven by.
Interesting. Why do you think that is?
I don't know. I'm doing the same place a little bit.
Yeah.
There it is.
So, but it's in mice, you said?
Yeah.
That you work? So you see the same thing in mice. That's interesting. Okay. Huh. Yeah, we've not done a lot of work on, I know that there's a large, we were chatting a lot at lunch about sex differences and hormone stuff. And we haven't, I mean, we're starting up a study in a transgender cohort looking at the role of hormones, but we haven't done a lot of stuff so far on that. But yeah,
Sorry,
interesting. Yeah.
question. Could it be associated with the heterogeneity in your clinical sample? So because you have a lot of differences within that sample, then you only detect regions that show change in very, very severe cases, but not in very mild cases that are close to your controls?
I thought what you were going to say was more about like the retest reliability of the imaging data, because I do think that might have something to do with it, that there might be just less variance across people as a whole in like these earlier somatomotor cortices than in these higher order cortices, and that, you know, if you just have less variance, then you can get you have better SNR to get an association. I'm not sure about, I mean, we see the same thing in the very first, one of the very first pictures that I showed you with all of the thing that wasn't legible, right? With all of the different red bars in the CCA. Actually, some of the strongest things are in, and that was with 20,000 people or something.
Yeah, I think for me, it's exactly your point, but reverse. So because the effect is in, the effect is initiated in different regions, but you can only detect it in regions where your subjects are at a severe stage. So like, if it's not associated with visual cortex, let's say the amygdala, so then you only detect it in the visual cortex because your cases that are severe, your effect is spread to that. So at that stage, when it's really a severe case, you will see it in regions where your sample, most of your sample would have that.
Yeah, well, but most of our sample is not that severe. And we have done things where we sort of subsample based on severity. So we start an analysis based on 20,000 people, and then we systematically go more and more severe. It's like smaller sample size, but more and more severe. And I think my memory is that you see this pretty, that you see this across the board, regardless of where it doesn't really change. But it's interesting. I mean, I've also been thinking about the kind of sensory motor association axis is maybe if something changes on one kind of side of that axis, there's like an echo of it on the other side. And so it's just a sort of, especially given that this is trait, it's just like a developmental system in terms of how these are neurobiologically not independently developing of each other. But yeah, we're hoping, well, we first of all want to do this meta-analysis to really confirm that this is a robust finding and kind of get it out there. And then we want to start doing some more comparative stuff to come up with different hypotheses and try and see if we can see what might be explaining this. But I'll look into the sex difference side of things. I hadn't thought to look. I think that would be interesting to see if it's the same in people. Okay, so that's all I was going to say on that study. So the 2 more studies to go. If we don't get through it, it's fine. I would rather, have these sorts of discussions. I want to talk a little bit about comorbidity. So this is the study that we talked about a little bit at in our lunch. So the goal here was to classify using machine learning model using brain imaging features, multiple diagnosis. This is work led jointly by Tai Easley and Lexi Liu in the lab, and this is now out in GigaScience actually. And so the reasons for this was, so we went into the ICD-10 codes in the UK Biobank, and we selected 17 different diagnostic categories that had, just based on whether they have enough patients. We had a threshold of I think 120 patients. And then again, we had completely matched age, sex, and head motion matched controls, the same number in each case. And we wanted to do this partly because we could, it's when you've got a sort of population data set, it's a unique opportunity that other data sets don't afford you. But really what I wanted to do is like train a model on one disease and then apply it to another disease and see which sort of which modeling predictions generalize across diagnostic sort of boundaries and which don't. We didn't do that, and I'll show you why. So this is, I'm going to show you the results in a minute. So these are the 17 different sort of diagnostic categories on the horizontal axis. prediction accuracy on the vertical axis. Chance is always at 50% because we always have the same number. We just have a matched, no class bias, so a matched patient and controls. And so these are the results. There's distributions because we did shuffle split, so essentially repeating so that you can get the variance as well as the point estimate of the accuracy. In color is the best performing feature set, and in gray is all of the feature sets. So we had a few different structural MRI feature sets, so parcellated cortical thickness, volume, those things. We had quite a few different functional data feature sets because we were kind of interested to see whether some types of features might predict some diseases better than others. We also threw in the end a sort of demographic and socioeconomic feature set because we were like, maybe that will help, because essentially the takeaway is that these are not that good. So they're all sort of better than chance on average, but the only ones that are really robustly and significantly better than chance are these two. So it's depression and demyelinating diseases, which includes MS. This is kind of a cool statistics 101 because power is the combination of your sample size and your true underlying effect size. And these two things that are significant are actually the largest group and the smallest group. And so the largest group is significant not because the effect size is that big, but because the sample size is big, and so we get less sampling variability, and so you get a p-value that meets your threshold. The demyelinating diseases is the smallest one, so it's not significant because we have a big sample, but it's the furthest away by far, so it has probably the largest underlying effect size. So even though you get a large variance, you meet that significance. So it's kind of cool from that geeky perspective. But yeah, we were a little disappointed about this, and we can get into it in terms of like all the things that are wrong with the ICD-10 that are probably explaining some of this. Anyone want to? I know we chatted about this. There you go. I knew you were going to have a question.
And during the lunch and this kind of thing to read it. I found it very interesting, but I did have like another question. So like a lot of these diagnoses are used for people that like comes like mononeuropathies, polyneuropathies, things like carpal tunnel syndrome, things like that. So I would argue that I would actually be surprised if you would find like large associations with cortical gray matter volume and carpal tunnel syndrome, for example. So would you, like from that point of view, like would you say that like it's only the problem is only with the ICD-10 code, or it's like the specific input features that maybe you're using could be related to like some diseases more than others?
Yeah, I think that's totally fair. We went for chapters F and G in the ICD-10 mainly because we didn't want to handpick too much. We want to have some, and F and G are, one is mental and one is neurological. So we were like, oh, those are brain words. And then we selected based on sample size beyond that. So we wanted to not necessarily exclude beforehand, but sure, it makes sense for some of these to not be super predictive. We had hoped that, movement disorders, anxiety, and a lot of other things would be. And then we wanted the original idea was then to swap predictive models to see whether the model that you trained on anxiety could also predict depression, for example. But we didn't even end up trying that because most of these models don't even do that well in their own samples. Yeah.
Very related to that. Again, like kind of more common measures into your input features are like functional, but they do look also at the microstructure and more white matter features. That could be really the interesting person of these like stroke and MS.
I don't think we did for this. I mean, the initial interest, this was a project that was partly overlapping with our kind of resting state functional methods comparison kind of angle of the lab, and that's Ty, who is the first author on this, was the person who kind of mainly designed this project. And so one of there was the sort of swapping models was one of the research questions initially that kicked off this project, and the other was seeing whether some functional things are more predictive than other functional things. So actually, that's all it turns out there's no difference basically. That didn't even make it into the main text of the paper, but that's sort of the historic background of how this project evolved. So we didn't look at. I think that'd be interesting. Yeah, there's certainly more clinically important features that one could look at. Yeah. Thanks. How are we doing for time? What time is it supposed to end? 3? Yeah.
The structural doesn't include voice over other intensities.
I think that's right. I think we focused on, so the question was, the structural does not include wider hyperintensities. I believe that's right in this case. I believe we focused on,
What that could be helps with
maybe I should double check. I'm pretty sure we only use the sort of T1 derived cortical thickness and structure and volume.
That'll be messed up by the hyperintensity, so you'll get a signal.
Yeah, so right. Not independent. Yeah.
Firstly, I share your, like, I wish it works.
Thank you.
So, but even with these sample sizes, is it possible that we are getting lost in the high dimension? Like what, because it's very difficult. Like in the one hand, yes, we are not finding what we expect, but on the other hand, these things are happening in the brain.
Yeah.
And we are not finding them. So
I know, that's so frustrating, isn't it?
So they're probably missing these things that are happening in the brain.
Yeah. So with high dimensional, you mean that
I mean, like, we are not. So do you think if we had, instead of 2,000 for each of these 200,000, then things will start emerge, or is it like the effect sizes are so small in each part, or what is happening? Essentially, that's my question.
I mean, I think if we had 2,000 in each group, many of them would be significant, because if you look, these thick dots are like the medians, and the depression level is similar to a lot of them, right? And it's only significant because we have a bigger sample. Can you give me
Ignore that it's not significant. Go with the effect size and go around and be like,
and still do it?
and still do it, because they do it in genetics. They're like, no, they do it. It's like you're correct for things. Sometimes they're like, what if we just get the effect sizes and we get a PDPRS, ADPRS? Like,
Yeah.
the accuracy is like 50-something%.
I know.
For most of them.
Yeah,
But
but you're
I'm
saying,
saying, what if the accuracy is 50-something% for itself, but it's also 50
but also
something
for something
if you
else.
use another disorder. If like, because based on this, you expect the accuracy from the other one will be like 50%. Right?
Or even worse. Yeah.
But if it explains the same amount, and if you can cluster them across the G's and
Yeah.
H, maybe there is something. It's just the effect sizes are smaller. And why would we punish it if we didn't have the sample size? Yeah, I'm like, I hope it goes through, because it's very.
I like it. You're telling me to still do it. Yeah, no.
Because I was like, I hope that it will work. And my
Yeah.
next question was like, oh, does this also correlate with those small effect sizes and genetic correlations between things? Where does this come from? But I see that.
Yeah.
I'm still like, how can we approach? Because we know, for example, epilepsy, these things are happening in the brain. And if you really target, as you said, more severe cases, or maybe we will get some signal. And if we are not getting any signal, then the question becomes like, what is happening? Is it just, it's all neurochemical that we are missing with MRI machine? Or
Yeah, someone yesterday was telling me that this sort of thing might be doable with Enigma data, because there's multiple different clinical Enigma cohorts, and so you could do this sort of thing there. And maybe it's also big, at least big-ish, and so
I should say that I think this is my person. Your data is cleaner. It's like all the same protocol. Enigma
It's
is.
the first time anyone's told me that.
I haven't worked directly with Enigma.
Yeah.
Getting the data, but
No.
you get summaries that
Yeah.
come from different scanners, and it's like it's a lot more.
Right, right. Yeah, in terms of this is very harmonized in terms of the imaging. That's certainly true. Yeah. Yeah, maybe we should try. I mean, you're right that even though, I mean, and we did have pretty rigorous statistical thresholds, like you could come up with a different way of testing significance that might make 6 of these significant. I mean, at the end of the day, I don't care that much about p-values because you learn not to when you work with the UK Biobank. And it's but you're right, like maybe we should go back and still try and see if because you could get similar kind of distributions, right? And even if it's not significant, it would still tell you. I think the because we did the opposite. Obviously, I can't suddenly have 2,000 in each group, but we did do the opposite in response to a reviewer, which is to downsample each group to the smallest group and essentially match it, do H and 6 matching to the smallest group. And so you get the distributions become equally wide for each of them, of course, because you get the same sampling variability for each of them. But the middle dots stay pretty much, it's pretty much unchanged from this. So I think the central points are reliable estimates of how good these models are. So yeah, to your point, we could do that. Yeah. Okay, so I'm going to talk about one more project that actually only just came out. This was again work from Kayla Hannon. This was a registered report for open science people. This was my first registered report and I loved it. I'm going to try and do it more often. This is where the pre-registration is where you submit your plans to like a repository. Your registered report is where it actually gets reviewed. And a journal says, we will, if you do exactly what you say you're going to do, so you submit the introduction and the methods, it gets reviewed by reviewers. And then the journal says, if you do what you say you're going to do, we're going to accept it regardless of the outcomes, the results. And I think it was good because this project would have died had we not done that beforehand. And you'll see why probably in a little bit. So here the idea, essentially the question is, are subtypes any good? And are subtypes helpful? And we, had looked at we were really interested in depression heterogeneity. We had looked into the literature. There's lots of, high impact, really cool papers that do data-driven subtypes in depression. And several of them show, treatment effects and stability and these other things. But when you start to look at multiple papers, you're like, wait, they seem to get like different numbers of subtypes and like different characteristics of subtypes. So the goal of this project was to essentially compare pre-existing subtyping approaches, but in the same cohort. So what Kayla did is she selected these, so each column here is a different paper, and she selected these 6 different papers and tried to replicate as closely as possible in the UK Biobank. This is not intended to be a replication study because these are not, we didn't prioritize the we replicated as closely as possible, but the priority was to keep the comparison fair. So for example, we didn't pre-process the neuroimaging data the same way the original paper did, because that would actually create potentially some differences between these things. So the priority was the comparison. And so we also had exactly the same inclusion criteria across all 6 so that we had the same cohort to work on. I'm not going to go into the details of this. I'm happy to spend more time in this. Basically, 2 of them are data-driven approaches that work using clinical data, like self-reported item-level questionnaire data. 2 of them are using structural MRI features. One of them is like regional gray matter volumes. The other is like a structural covariance sort of network inputs. And then 2 of them use resting state functional MRI. One of them is on time courses, so using dynamic information, and the other is on parcellated functional connectivity matrices. And so essentially we applied all of these 6 approaches as closely as possible to the original in the same like 2,000 subjects from the UK Biobank. And then we compared them, and we compared them in a few different ways. And I'm showing it this way around in a minute. So the first thing, well, this was not actually the first thing. This was one of the later things we did, which we might not have done had we not submitted this as a registered report. So the one of the questions we asked were, are these things robust? So if within one method, if you bootstrap and do it over and over again on different kind of subsets of the population, how similar do you get? How internally consistent are these subtypes that we're getting? And there's some interesting variation going on here, but overall, this is pretty good. You know, these are relatively stable. Most of them are significant, except for the resting state plus ward hierarchical clustering. That's probably because of an issue with hierarchical clustering. It's pretty ordering dependent, and so it tends to be less stable. But but this is pretty good. This was encouraging. These things are actually more stable than I might have predicted that they were going to be. And that's maybe partly because we had a pretty large sample size here. You know, you might get more variability. Some papers have shown more kind of instability in smaller sample sizes. The other thing we asked is, do they mean anything? Are these the resulting subtypes that we're getting, are they sensitive? And so these are some different kind of types of variables. None of these variables were included as features in any of the 6 approaches, although some are conceptually similar, like the mental health one. Those don't include any questions that were used as symptoms, but they might, or as features, but they might include questions that ask sort of similar flavor of information. And so the effect sizes are small. We're used to this by now, but actually these things, you know, there is some sensitivity, and unsurprisingly, the 2 blue ones are the ones that are clustered on clinical information, and they are the most sensitive to different but conceptually similar sort of mental health features. So the answer is mostly, yeah, these subtypes seem pretty robust and interpretable and meaningful. And so then the most important question, the first thing we did after we had applied done this work, was, do we actually get the same subtypes? Like, do these different approaches result in groupings that assign patients in similar ways? And this is a plot of adjusted rand index. You don't have to know that much about adjusted rand index. Basically, 0 means that no, there's no agreement whatsoever above chance, and these numbers are essentially 0. And so you get completely different subtypes. This was worse than I had expected. I thought, you know, if you get 2 different clustering approaches based on like clinical information, maybe they'd agree, right? And then if you get two approaches based on structural information, even though they're, you know, different clustering methods, slightly different features, maybe they would agree, but you don't see any sort of block diagonal structure either here. So there was less agreement than we had expected. Yeah, go ahead.
So these subjects, how many are there?
There's at least 2,000. I think about
UK
around
Biobank.
2,000. UK Biobank, depression. Yeah.
You can still color them with the same.
Yeah, actually, I have a brainstorming meeting with a couple of students, including Kayla, next week when I'm back in St. Louis, to think about doing something like that. So to think about combining the 2 approaches and seeing.
This
Yeah.
is very interesting.
Mm.
And the question is, like, the first study, you're addressing one of the main issues that
Mhm.
the heterogeneity. And then we go here, like, clustering is as good as what we can do.
Right.
We put a label, but if it's really heterogeneous, each of them are capturing something real based on previous slide.
Yeah, I agree
But
with you.
not the whole picture.
Yeah.
Maybe because there's so much to capture that.
Yep.
Yeah.
Yeah, so we're looking into whether we can do that. Even when you've got a big data set, you sometimes have to worry about sample size, because the more squirrely you get in terms of like splitting people up, the smaller it's going to get. And so, yeah, but I think it's an interesting point, and we're going to look into that for sure. Yeah.
This gives us hint.
Yeah. Well, so I have, I think, one more slide. So no, they certainly do not agree. So this was all of the stuff that we had pre-registered. We did this first, and if we hadn't pre-registered the study, we might have stopped here and been like, well, that was a waste of time, but we were like, well, we told them we were going to do all of these things. They're going to publish it regardless of what we find. So let's do the other things as well. So we were kind of encouraged by these findings to be like, oh, at least our paper will have something like interesting and valuable to say, to be like, this is not all garbage. It's just like you say, capturing different sides of things. So one thing that we did when you do a register report, you do what you say you're going to do. You can add on additional things as long as you say they weren't included in the original register report. And so we did do that because we were like, well, why is this, right? What is the reason why these things are so different? And we tested two different kind of data science flavors of that. So one reason why there might be a disagreement is because of the clustering method. You know, it's Ward's clustering, it's k-means clustering. These are different kind of methodological approaches. Or another might be because of the feature sets, right? Because the feature set are not quite the same across everything. And so we kind of did this big feed everything into everything, right? Feed each feature set into each of the different clustering methods. There's only 5 here because one of these clustering methods relies on time series data, and so you can't feed gray matter volumes into that. Doesn't make sense. But so this is kind of, these are averaged ARIs now across multiple kind of combinations of things. So when you feed the same clinical feature into all of the different methods, all of the different approaches, these ARIs are, I mean, they're not that great. They're not that close. You can go up to 1. They're not that close to 1, but they're not 0, right? You can, using the same feature set across different methodologies, you do get more similarity across clustering approaches. Whereas vice versa, if you have the same clustering approach, but this time feed different feature sets in it. So it's essentially the same data, but like averaging in different ways. Now you're getting back close to 0. And again, this was slightly surprising to me. I thought it might have been more to do with the method rather than the feature set, but the answer to this seems to be features. So it seems like even small variations, I mean, the 2 clinical feature sets, they have almost overlapping questions in terms of like, you know, how depressed are you feeling over the past 2 weeks? I think one of them included anxiety, one of them anxiety questions, one of them didn't, but they were they weren't that high dimensional, the features. I think there were like less than 50 features in both of those. And they were overlapping in terms of the questions, and yet they still disagree pretty strongly. Yeah, go ahead. You want to pass the,
Very, very
okay.
Sorry if I missed this. How many clusters are you using in this?
So the question is, how many clusters am I using in this? We let each of the original papers that presented these approaches had a method for deciding on the clusters. Some of them were based on bootstrapping, some of them were like the original elbow sort of method. And so we did what they did in terms of deciding.
So what about going along the dimensionality of number of clusters?
Yeah.
Because you're trying to compare overlaps across different methods with different number of clusters.
Yeah, that's totally fair. We did do that. So we fixed the number. I think we fixed the number of clusters to the median number of clusters that were optimized for each of them. And then we redid all of the comparisons, and it doesn't help. So you still essentially get 0, even if you have the same number of clusters across everything.
So I'm thinking about the method done here, but along the dimension of the number of clusters. So 2 clusters, 3 clusters, 4 clusters, and see, is there convergence, divergence? Is there places where are there a number of clusters that are more consistent across these other dimensions, features, or methods?
We did do some things that went like as a function of D, essentially, like number of clusters. I'm not sure we did this for that. So you think, why might that fix things? You know, if we have a fixed number of clusters across all of them and they still don't agree.
Is there a higher up in the tree where they do agree?
Oh, is there a higher up in the tree where they do agree?
Are they splitting in different places?
Oh, so you're not even saying like within the 2 cluster solutions, do they agree? And you're saying like, do some of the 4 cluster solutions agree with the 2 cluster solution somewhere else?
That would be complicated work.
Yeah.
Like 2 cluster solutions agree. Do they agree more or less at different levels?
We didn't do that in this paper. We are working on a newer project where we're perhaps foolishly developing a new method for doing subtyping, which is rather than on clinical or on neurobiology, we're clustering sort of on the relationship between the clinical and the neurobiology. And there we've done some of that, and it gets worse as you go into higher dimensionality. So you get even when you penalize for it, because the adjusted RAND index is adjusted for the complexity of the data. So my intuition is that it wouldn't, but we didn't explicitly test that here. That's a good idea. Okay, so these are some of the takeaways that I've talked about a lot already. So it seems pretty clear from all of this literature that exists already, and some of the work that we've done is that the brain basis of depression is pretty highly multivariate and spatially complex. And heterogeneous. And I think to the discussion we were having, there's not just one source of heterogeneity, there's lots, and we didn't even look at genetics, right? You can probably also do subtyping on genetics and get yet another set of meaningful, consistent, but non-agreeable clusters. And so I think my main takeaway from this is that it would be beneficial to do more comparisons. You know, in the machine learning literature, if you want to present a new model or a new architecture, you have to compare it against the performance of other architectures. And I think here, that would be a helpful thing to do, to at least like know to what degree newer subtypes are agreeing or disagreeing with previous approaches, so that we can at least, you know, build more of an integrated view of these things. And I think to some degree we need to embrace the complexity. You know, there are a lot of more complex models that are coming about, and you know, you want to use the simplest model possible, but I think we shouldn't shy away from trying to model and capture some of that complexity.
Possible.
Yeah, yeah. Well, we always, I mean, when you have the UK Biobank data set, there's sometimes a like just tendency to do the like let's chuck it all into one model. And we've certainly hopefully you've seen that we've tried to like be more intentional than that, and like ask more specific targeted questions than that. So when I'm saying embrace the complexity, I'm not saying let's chuck it all into one big thing and see what falls out, but like at least use the power that we have and the sample size that we have to sort of explore things in different directions. And so I'll just shout out a huge thank you to all of the collaborators, my amazing lab, and our currently still active, we'll see what happens, funding. Thank you for everything. Yeah, we had a lot of good discussion already. Appreciate it. It makes it so much more fun to like have some input whilst I'm not just talking at you. So yeah, I'll be around for a little bit. So come up if you want to chat more.
Thanks a lot. Thank you very much.
Thanks.